Good morning. I am Uday Kumar, working as Associate Professor in Department of Pharmaceutical Technology, Aditya Pharmacy College. I am going to take today my presentation is, I would like to give my presentation topic, Pre-formulation studies, physico-chemical characterization of drug substances. So, in this pre-formulation studies and physico-chemical characterization of drug substance, you know what is meant by pre-formulation? Pre means before, formulation. Before formulation, what are the studies are to be carried out, and what are the various physico-chemical characterization of drug substance. Drug substance is nothing but API, active pharmaceutical ingredient. Yes. When you know already what is the difference between API and drug dosage form. Once the API is mixed with the excipient, then you will get the dosage form, right? So before formulation, pre-formulation, what are the studies are to be carried out? That and all I am going to cover in pre-formulation studies. Mainly physico-chemical characterization, what are the various physical properties and chemical properties of the drug substance that are going to be affect the drug bioavailability, that I am going to cover in this presentation. Yes. So I am going to cover introduction to pre-formulation studies, what are the main objectives or goals of pre-formulation studies, and the study of physico-chemical characterization of drug substance. Under physico-chemical characterization of drug substance, organoleptic characters, bulk characterization, solubility characters, stability characters, yes, all those things I am going to cover. So pre-formulation, I said already, pre means before, formulation, before formulation, right? So it is defined as the phase of it is pre-formulation is one of the phases of R&D, research and development, in which pre-formulation studies characterize the various physical and chemical properties of drug molecule. Why we need to know the physical and chemical properties of the drug substance before the formulation? Means finally ultimate aim of our ultimate goal of our any formulation is to develop a safe and effective and stable dosage form. In order to get the safe and effective and stable dosage form, we need to characterize the various physico-chemical properties of the drug molecules. Before then only we will develop the safe and effective and stable dosage form. So next comes to the goals of pre-formulation. To formulate an elegant, safe and efficacious dosage form with good bioavailability. I already said ultimate aim of any R&D or formulation scientist is to formulate safe and effective dosage form, and it will give the good bioavailability. The drug provides that the drug will give good bioavailability when compared to the existing products. Yes. What is meant by bioavailability? Bioavailability is nothing but rate and extent of drug absorption that is going to be reaches to the systemic circulation. The rate and extent or amount of drug that is going to be reaches to the systemic circulation. Already one formulation or dosage form is there. Again we are developing the another same dosage form. Whatever the developed dosage form will shows the better properties than the existing one. When compared to the existing products, existing products, formulated products bioavailability has to be improved. Yes. So that is nothing but that is the main goal of any objective. goal of our pre-formulation studies. Why means already one formulation already existing in the market, just we are changing the excipients and we are changing the method of manufacture. Finally, we are going to get the better bioavailability than the existing products by modifying the physical properties, by modifying the chemical properties, by modifying the excipients, by changing the excipients, by changing the route of manufacturing. Finally, we are going to get the better bioavailability than the existing products. So that is the main goal of pre-formulation, to formulate an elegant, safe, efficacious dosage form with good bioavailability. I already said second goal, to formulate new dosage form, to formulate new dosage form of already existing drugs. I said by changing the excipients, by changing the method of manufacturing. So to formulate a new dosage form of existing drugs. And third goal is determination of all the properties of drugs and the best suitable dosage form for the drug molecule. Before formulation, we are going to study all the properties of the drugs, all the drug molecules. We study all the properties of the drugs. From that, we can select the best suitable properties. Then only we can develop the safe and effective dosage form. Understand? So I repeat, to formulate an elegant, safe and efficacious dosage form with good bioavailability, to formulate new dosage form of already existing drugs, determination of all the properties of drugs and the best suitable dosage form for the drug molecule. And comes to the objectives of pre-formulation studies: to establish physico-chemical parameters of a new drug entity. I already said we are going to be developed, or we are going to study the various physico-chemical parameters of new drug substance or API. And apart from that, second, to determine its kinetics. Kinetics means to determine the kinetics and stability. Kinetics means rate and velocity, how much fast, how much rate it is going to be delivered the drug reaches to the systemic circulation. That is called kinetics. And stability. Stability means whatever the product we are going to develop, how long it is going to be stable. Whatever the product we are going to be develop for a new drugs or new drug substance or new drug formulation, until its expiry, the product should be stable. The main objective of conducting pre-formulation studies is to establish, to determine its kinetic properties, as well as to establish the stability data. That means to know the suitable storage conditions, how much temperature and how much humidity we can store the drug product to maintain its integrity until its usage, until its expiry period. Then only if you are going to store the product properly until its expiry period, the product properties remain same, or the product properties never to be altered or never to be changed. That is the main objective, to determine its kinetics and stability, to establish its compatibility with common excipients. Because I already said, if you want to formulate any dosage form along with the API, you require excipients. Yes, even though API only produces the pharmacological action, excipients also plays an important role for getting the good, efficacious, safe product. Because any formulation we are going to add various excipients, n number of excipients. Even though you are going to add n number of excipients, every excipient will having its own function, every excipient is having its own usage. all the excipients are not suitable for the particular drug, because whatever the excipient we are going to use, the particular excipient will having compatibility towards the drug. What is meant by compatibility? If the addition of, because finally what is in formulation contains both drug and excipients. If the on addition of the excipients, whatever the excipient we are going to add, the particular excipient will having compatibility towards the drug. If the excipient does not having compatible towards the drug, what will happen? The properties of the drugs will be changed. The properties of the drug will be changed. Understand? So before addition of the excipient to the formulation, we are going to check the compatibility of the drugs and excipients. If the excipients shows reaction between the drug and excipients, the particular excipient is not suitable for the formulation. That is, particular excipient is incompatible towards the drug. That means on addition of this excipient, drug properties will be changed, will be altered. Yes. But so how will you know this whether the particular excipient will having compatibility towards the drug or not? By conducting FTIR studies, Fourier transform infrared spectroscopy. Fourier transform infrared spectroscopy. The functional groups, position of the functional groups will be changed, if at all, if any reaction can occurs between the drug and excipient. If the reaction cannot, any reaction cannot occurs between drug and excipient, the functional group position cannot be altered by using FTIR. FTIR scans, FTIR Fourier transform infrared spectroscopy spectras. We know the functional groups positions. They based on that we can determine particular excipient is compatible or not. That is the main compatibility, the main goal of or I can say main objective of any pre-formulation studies. Understand? To establish its compatibility with common excipients, and last objective is it provides insights into how the drug product should be processed and stored to ensure their quality. Where we are going to be stored the particular drug, because all the drugs cannot be stored in same storage conditions. Based on the drug properties, drug molecules nature, we are going to select the different storage conditions. I am repeating, I am going to conclude these objectives. The main, any pre-formulation studies main objective is to establish the physico-chemical parameters of a drug entity, to determine its kinetics and stability, to establish its compatibility with common excipients, to provide insights into how drug products should be processed and stored to ensure their quality. Comes to the outline principal areas of pre-formulation research. I already said pre-formulation is one part of R&D, right? Like pre-formulation, formulation, analytical development, preclinical, clinical, like that. But all the stages before the formulation, we are going to study the pre-formulation. That is so. That is the reason pre-formulation is very important step. One of the part of research R&D. Yes. So outline of principal areas of formulation research. Mainly physical. I already said in earlier slides. I already explained. We are going to establish various physicochemical parameters of the drug substances. We can characterize, we can analyze, we can understand the various physicochemical characteristics of the particular drug substance. Like there are three properties we are going to be mainly we are going to focus: physicochemical properties, bulk characterization, and solubility analysis and stability analysis. So principal area, there are four principal areas like physicochemical properties, bulk characterization, solubility analysis and stability analysis. Under physicochemical properties, mainly purity of the particular drug substance. Yes, purity means without any impurities. And surface area, organoleptic properties like color, odor, and particle size and particle shape. So these are the various physico-chemical properties like organoleptic properties, particle size and shape, purity and surface area. And bulk characterization, under bulk characterization we are going to study the crystallinity of the drug substance as well as polymorphism, hygroscopicity, particle size characterization, bulk density, powder flow properties like, and understand I am repeating: bulk characterization, crystallinity and polymorphism, hygroscopicity, particle size characterization, bulk density, powder flow properties. Under solubility analysis, we are going to study the ionization constant, pH solubility profile, common ion effect, thermal effects, partition coefficient, solubilization, dissolution, right. So under solubility analysis, I am going to repeat various properties we are going to be characterized like ionization constant pKa, pH solubility profile, common ion effect, thermal effects, partition coefficient, solubilization, dissolution. Last one, stability analysis. Under stability analysis, solution stability, solid state stability, bulk stability, compatibility, bulk stability and compatibility. So these are the various 4 properties like physicochemical properties, bulk characterization, solubility analysis and stability analysis. These main 4 areas mainly we are going to be studied under pre-formulation. So first one is organoleptic characteristics like color, odor, taste of the new drug substance must be recorded. Yes, because any drug substance will comes for analyzing. First parameter we will see this color. Yes, color of whether the particular drug substance is which color, of white or creamy yellow or shiny. Any what in the beginning of analysis, if the description itself fails, means straight away we are going to reject the product. Understand? No need to go for further analysis. So description is very important. So we need to examine the description very clearly. Based on that, we can determine we can go for the further analysis or not. Yes, understand? So organoleptic character, even though this property looks very simple, but is very important from the pharmaceutical industry point of view, because almost all the drug substances are looks very similar, right? White, off-white, creamy white. Very narrow difference will be there. So we need to observe the description. We need to see the description very carefully, thoroughly. Yes, color, odor, taste of the new drug substance must be recorded. So various colors like off-white, creamy yellow, tan and shiny, and odor, pungent, sulfurous, aromatic odor and odorless. Taste, acidic taste, bitter taste, intense, sweet, tasteless. So based upon the drug substance, the odor will be different, like aromatic, odorless, pungent. Based on the taste also, different tastes will be there: acidic, bitter, sweet and tasteless. So these are about outline properties, outline characters of any organoleptic characteristics like color, odor, taste. And next comes to the bulk characterization. First one is crystallinity. What is meant by crystallinity, right? Based on the internal structure, right? For crystal habits and internal structure of drug can affect the bulk and physicochemical property of molecule. What is meant by crystal habit? Crystal habit is description of outer appearance of a crystal. Outer appearance. And outer appearance is different and internal structure is different, right? So outer appearance, that is nothing but crystal habit. Internal structure of the drug can affect bulk and physicochemical property of molecule. Crystal habit is description of outer appearance of crystal. Internal structure is molecular arrangement within the solid. Internal structure is molecular arrangement within the structure, right? If change in the internal structure, within internal structure usually alters the crystal habit. Change with internal structure usually alters crystal habit. Conversion of sodium salt to its free acid form produce both change in internal structure and crystal habit. Conversion of sodium salt to its free acid form produce both change in internal structure and crystal habit. So these are the various different shapes of crystals: cubic, tetragonal, and triclinic, orthorhombic, hexagonal, monoclinic, trigonal, right? Understand? See, I am repeating, crystal habit is nothing but it is description of outer appearance of crystal. Internal structure is molecular arrangement within the solid. Change with internal structure usually alters crystal habit. Conversion of sodium salt to its free acid form produce both change in internal structure and crystal habit. So already showed the various crystal forms, right? Crystal shapes like cubic, tetragonal, triclinic, orthorhombic, hexagonal, monoclinic, trigonal. Next comes to the different shapes of crystals depending on internal structure. I already said crystal habit is nothing but outer surface, and depending on internal structure, compounds is classified as crystalline and amorphous. Yes. What is the difference between crystalline and amorphous? Crystalline means proper structure, amorphous means that does not having proper structure. Understand? Depending on its internal structure, compounds is classified as crystalline and amorphous. Crystalline compounds are characterized by repeated spacing of constituent atom or molecule in three-dimensional array. Crystalline compounds are characterized by repetitive spacing of constituent atom or molecule in three-dimensional array. So that means like by orderly they are going to be placed. Understand? Repeatedly spacing of a constituent atom or molecule, they are going to be placed in a specified manner. Whereas amorphous form or molecule, they are going to be placed in a randomly. They are going to be placed very randomly, zigzag. Whereas crystalline, they are going to be placed in a orderly. Understand? That is the reason I already said, depending on internal structure, compounds are classified as crystalline and amorphous. Crystalline compounds are characterized by repetitive spacing of a constituent atom or molecule in three-dimensional array. In amorphous form, or atom or molecule are randomly placed. As far as stability is concerned, as well as stability and dissolution rate is concerned, the amorphous forms are more for stability as well as dissolution is very greater for amorphous than crystalline form. Yes, so obviously when the drug is available both crystalline and amorphous, we are going to select the amorphous form because of the solubility as well as the dissolution parameters. So solubility and dissolution rate are greater for amorphous form than crystalline, as well as amorphous form has higher thermodynamic energy. Example, amorphous form of novobiocin is well absorbed, whereas crystalline form results in poor absorption. Understand? So if the drug is available in both crystalline and amorphous form, in pre-formulation we are going to study the various properties of drug substance, whether it is available in crystalline amorphous form, which form is stable, which form having more solubility, which form having more dissolution rate. Finally, we are going to select the more stable, the drug which having more solubility, the drug which having more dissolution rate. That form only we are going to be select for our formulation point of view. If you are going to select other one, the drug is not stable, the drug does not having proper solubility, the drug does not having any dissolution. So finally the drug cannot produce any pharmacological action. Understand? So that is the reason we are going to study the various properties. Finally we are going to select the desired one, like solubility, dissolution rate, yes, and greater, both solubility and dissolution are greater for amorphous form than crystalline. So amorphous form has greater thermodynamic energy. So it is the ability of the compound. Next comes to the polymorphism. It is ability of the compound to crystallize. Poly means many. Same compound will be available as different forms is called as polymorphism. It is ability of the compound to crystallize as more than one distinct crystalline species with different internal lattice. Understand, different crystalline forms are called polymorphism. Polymorphs are two types, enantiotropic and monotropic. Enantiotropic and monotropic. Understand, poly means many, morphs, same compound is available in different forms are called as polymorphism. It is the ability of the compound to crystallize as more than one distinct crystalline species with different internal lattice. Different crystalline forms are called polymorphs. Polymorphs are two types, enantiotropic and monotropic. Enantiotropic and monotropic. The polymorph can be changed from one form to another by varying temperature or pressure, that is called as enantiotropic polymorphs. Understand, example sulfur. One polymorph which is stable at all temperature and pressure are called as monotropic polymorphs. Monotropic polymorphs. So, can you now understand what is meant by enantiotropic and monotropic? Enantiotropic is nothing but the polymorph which can be changed from one form to another by varying temperature or pressure is called as enantiotropic polymorphs. Sulfur. One polymorph which is stable at all temperature and pressure is called as monotropic polymorphs. Example, glyceryl stearate. Last one, polymorphs will differ from the, I already said, same poly means many, morphism, same drug is available in different forms. Why? Even the different forms available, one form will be varied to another form with respect to their properties. Yes. So we are going to study all the properties. Finally, we are going to select the desired one, like solubility, melting point, density, hardness, compression characteristics. So we are going to study all the properties of the different forms. Finally, we are going to select the good, high stable one, highly melting point, density, hardness, compression character, the forms which is having more superior characteristics. We are going to select the desired form for our formulation purpose. One example I am going to give here, chloramphenicol is available A, B, C, but B form is more stable when compared to A and C, so better we are going to prefer B form. Understand, poly means many. The forms, why? Even though same drug is available, same substance is available in different forms, we are going to select the more stable form for our formulation purpose. So how we are going to select the form? We are going to study all the properties. Finally, we are going to select what are different properties. Even the different forms are available. One form will be varied to another based on their properties like solubility, melting point, density, hardness, compression characteristics. Chloramphenicol A, B, C is available, but B form is more suitable. Why it is stable and highly preferable. Okay.